Hello and welcome to Sam Biology class. Friends, today we shall discuss about the principle, procedure and the applications of affinity chromatography. Affinity chromatography. Before that, I request all of my friends, please subscribe my YouTube channel and watch and like my videos and also share them with your friends. All of us are aware that an enzyme binds with a specific substrate or a group of substrates to form the enzyme substrate complex. To form the enzyme substrate complex. Similarly, a hormone also interacts with a specific receptor to form the hormone receptor complex. To form the hormone receptor complex. Similar interaction also takes place between an antibody and an antigen that result in the formation of antigen antibody complex. Antigen antibody complex. These are the examples of amazing biospecificity between an enzyme and its substrate, between the hormone and its receptor, and also between an antibody and its antigen. In these examples, substrate, receptor and antigens are called as ligands. Affinity chromatography is a powerful separation method that exploits this biological specificity. It utilizes the biological affinity of a macromolecule for its characteristic ligand. Affinity chromatography is performed usually in column mode. For example, we have a sample containing hundreds of different proteins from which we want to purify these particular enzymes. To purify these enzymes by affinity chromatography, a column is first packed with a suitable support medium such as agarose. After packing the column, the supporting matrix of agarose gel beads will be coupled to a substrate analog that will bind to the particular enzyme. When the sample solution is applied on the top of the column, various molecules in the sample will move downward. During downward movement, because of specificity, these particular enzymes that we want to purify will bind to their substrate analog. In this case, the substrate analogs which have been immobilized on the surface of gel beads serve as ligands. Because of this binding, these particular enzymes that we want to purify will be retarded. Additional molecules present in the sample will not bind to the substrate analog and hence they will pass down and come out of the column. After additional molecules have been flushed out of the column, the wash solution is altered so that these enzyme molecules are dissociated from their substrate analogues and after which they can be easily eluted out of the column. Similarly, antibodies can also be separated from a sample by immobilizing specific antigens as ligand. Based on the same principle, hormone receptors can also be purified by using specific hormones as ligands. Affinity chromatography is not limited to the proteins only. It is also useful for the separation of nucleic acids, cell fragments, and even whole cells. Next, the supporting matrix. A suitable supporting matrix used in affinity chromatography should have the following characteristics. Number one, the matrix should be innate to other molecules in order to minimize non-specific adsorption. Number two, it should have good flow properties. Number three, the matrix should be chemically and mechanically stable at varying pH, ionic strength and denaturing conditions that are often employed in elution and binding. And number three, it should contain a large number of suitable chemical groups for ligand attachment. And finally, the matrix should preferably be highly porous. In practice, particles that are uniform, rigid and spherical are used. The most commonly used ones are agarose, polyacrylamide and controlled porosity glass beads. Of these three, agarose is by far the most often used supporting matrix because it possesses most of the desirable features that were just discussed. Next, ligand. Ligands are selected after careful consideration. Substrate analogues, substrate analogues, effectors, enzyme activators, receptor antagonist or synthetic agonist, and antigen epitopes are the usual candidates. It is not usual to utilize substrates as ligands in affinity chromatography, but when it is the only choice available, the conditions are adjusted so that the enzyme substrate complex is formed but the reaction do not take place. Next, ligand attachment. Attachment of ligands to the supporting matrix involve two steps. First step is the activation of matrix functional groups, and second step is the covalent attachment of ligands to the activated groups of the supporting matrix. A ligand should meet two important requirements. Number one, it should interact strongly with the desired macromolecules, and number two, it should possess a large number of suitable functional groups that could be modified during covalent linkage with the supporting matrix. When a ligand is directly attached with the activated groups of the supporting matrix, macromolecules might experience steric restriction due to which they might not become able to adsorb onto the matrix. This difficulty is overcome by introducing a spacer between the matrix functional groups and the ligand. The spacer is known as the spacer R. Due to the presence of the spacer, the ligand is projected at a distance from the gel beads and the macromolecules become fully able to bind to it without facing any steric hindrance. Large variety of spacer arms have been investigated. All of them must possess two functional groups, one of which will react with the activated group of the supporting matrix and the other to which the ligand could be attached easily. The procedure for affinity chromatography has many similarities with other forms of liquid chromatography. Like gel permeation method, here also the gel beads are first swollen and then loaded onto the column. The buffer that encourages the absorption of macromolecules on the gel surface should be used. It also supposes high ionic strength in order to minimize the non-specific absorption of polyelectrolytes onto the charged groups of the ligand. The buffer solution also should be supplemented with cofactors such as metal ions which may be required for ligand macromolecule interaction. The sample is applied at the top of the column and then the buffer is passed through it. After binding of the macromolecules with their characteristic ligands, the column is eluted by using more buffer in order to remove non-specifically bound unwanted molecules. After all unwanted molecules have been flushed out of the column, macromolecules that are bound to their characteristic ligand are eluted by two separate methods. One is non-specific elution and the other is specific or affinity elution. Non-specific elution is carried out by changing either the pH or the ionic strength of the buffer solution, whereas specific or affinity elution can be carried out by adding molecules which compared to ligand has a greater affinity toward the desired macromolecules or by adding molecules which compared to desired macromolecules has a greater affinity toward the ligands. Affinity chromatography has many applications. Number one, in order to study the gene expression, molecular biologists frequently have to isolate messenger RNA from a sample containing different types of RNA. Affinity chromatography is routinely used for this purpose. This technique is based on the fact that only mRNA molecules possess a poly tail at their 3' end. These poly tails bind to the oligo-DT ligands which are immobilized on a suitable supporting matrix. In this technique, oligo-DT ligands are immobilized on the agarose matrix. When the sample is passed through such a column, only mRNA molecules, because of the presence of the poly tail, will bind to the oligo-DT ligands which are immobilized on the gel bits. Because of this binding, only messenger RNA will be retarded, whereas other types of RNA which do not have the poly tail will not be able to bind to the ligand and hence they will pass down and come out of the column. After removal of other types of RNA from the column, mRNA molecules that are bound to the oligo-DT ligands are eluted. Number 2. Affinity chromatography has also been used for the separation of DNA binding proteins. Number 3. This technique is useful for the separation of enzymes, immunoglobulins, membrane receptors, nucleic acids and even polysaccharides. Number 4. Metal-chelate chromatography is a logical extension of the basic principle. Many proteins that have similar molecular weight and even similar isoelectric point cannot be separated by powerful methods such as gel permeation chromatography. These proteins, however, differ in their ability to bind with specific metal ions. This property can be exploited in the separation of these proteins from a sample. And number 5, immobilized enzymes is another extension of the affinity principle. In this technique, enzymes are immobilized on gel beads that are packed onto the column. The substrate is then prepared in a suitable reaction mixture. This reaction mixture containing substrate is passed through one end of the column and the products are recovered from the other end. Friends, this is all about the principle, procedure and different applications of affinity chromatography. For more updates, subscribe my YouTube channel, Sam Biology Class.